those that got delta-9 THC or those that got delta-9 THC, the same amount, but all the other chemicals lost body weight. Those obese mice became lean, almost down to the level of the lean mouse body weight. So we run these tests and we find, despite that body loss, the mice that were given just purely delta-9 THC, they were still diabetic. However, the mice that received the whole extract with THC and all the other chemicals completely reversed that diabetic phenotype.
Welcome to High Five with Dr. Igor Grant, Distinguished Professor of Psychiatry and Director of the Center for Medicinal Cannabis Research at UC San Diego. Each episode will take you on a fascinating journey into the evolving world of medicinal cannabis, as Dr. Grant poses 5 thought-provoking questions to his guest, uncovering their personal journey and unique contributions to the field. Now, here's your host, Dr. Igor Grant.
Today, I'm very pleased to welcome Dr. Nicholas DiPatrizio. Dr. DiPatrizio is Professor of Biomedical Sciences in the School of Medicine at UC Riverside and founding director of the UCR Center for Cannabinoid Research. Dr. DiPatrizio's work focuses on how the body's cannabis-like signaling system helps regulate appetite, metabolism, and reward, and how problems in that system may contribute to obesity, type 2 diabetes, and related conditions. He also examines how cannabis exposure affects this system with the goal of identifying safer ways to treat metabolic disease. Welcome, Dr. DiPatrizio.
Thank you, Dr. Grant, for inviting me. This is quite the honor, and happy High Five Friday.
And same to you.
Yeah, thank you.
So before we start, would you say a few more words about your background and how you became interested in studying cannabis? Perhaps say something about your lab at UC Riverside.
Sure. Yeah, so it's kind of funny. I can pinpoint the exact moment when I became very interested in cannabinoid research. In fact, I was a very young man at the time. I was a junior at Temple University. It was 99, 2000. And I saw there was a picture on a posting on the wall in a psychology class I was taking with Dr. Toby Yarby. He's since passed, a very well-known behavioral pharmacologist. And I was taking one of his psychology classes, and it said on the wall, we would like research students to help out with a research project. And I thought, well, that sounds really cool. Let me talk to the professor. So I grabbed him after class and I said, Dr. Yarby, what is this all about? And he started to tell me about all the research he was doing, where he would get different pharmacological drugs, THC, and see how they would interact with other drugs, other antagonists, for example. He worked heavily with Dr. Makarianis at Northeastern Center for Drug Discovery. And he sort of dangled the carrot for me and said, you can help work in the lab and you may even get publications out of this. So I thought as a young man, like, wow, this is the best thing ever. Publications, like a real scientist. And from that moment, Igor, I was hooked. And from then all the way up through now, the common thread throughout all my research has been the endocannabinoid system and how cannabis impacts these pathways. And we've expanded now into looking at really from head to toe how the endocannabinoid system functions in the body under normal physiological conditions, how it becomes dysregulated in, as you mentioned, a disorder such as metabolic syndrome, obesity, type 2 diabetes. And we look at one major project in the lab is looking at how the brain talks to the organs in the body and how we have this bidirectional communication. So it's been a fun journey with that common thread of cannabinoid research. And I credit Dr. Yarby with getting me hooked on that.
Well, fabulous. We will get into the brain talking to the gut and back and forth a little bit later, I'm sure. But you mentioned the endocannabinoid system. Now, many listeners may be familiar with that, but may not understand what it has to do with the gut. Could you please briefly describe the endocannabinoid system and say a bit about how it specifically relates to function in the gut?
Yeah, right. So this is a system that's in our body, as we know. Again, it's from head to toe, almost in every single cell, regulating almost every physiological function within the body in some manner. So how I like to think of the endocannabinoid system is from the cannabis perspective. In order to understand how cannabis works, however, we have to understand how the endocannabinoid system works in our body. So as we know, cannabis hijacks this natural system in our body and overactivates it systemically within the body.
Hijacks it or just rents it or gets on the bus.
Well, it may rent it for a little while and then it gets metabolized, right? Right. So we know this system's located in the brain and throughout all organs in the body, including the gut. Now, the gut, I'm a trained neuroscientist, so why am I even exploring and thinking about the gut? Well, the gut, as you may know, is known as the second brain. It has 5 times the amount of neurons in it than does the spinal cord. So this is a very important system that regulates functions such as peristalsis, so movement of food through the GI tract, the breakdown of nutrients, the absorption of those nutrients, and then pumping these nutrients into circulation for our survival. What has been shown over the years is that the endocannabinoid system is prevalent throughout the GI tract. Some of the first studies were showing that it's involved in gut motility. So, for example, activating the CB1 receptor system can lead to a decrease in peristalsis, so decreased movement of foods through the GI tract. Now, the CB1 receptors are located throughout cells in the gut, including the enteric neurons that I mentioned that they're very abundant, make up our second brain, so they can control movement. But they're also involved in other aspects of GI functions, such as absorption of nutrients, how the cells are sensing the nutrients. And this is an area that we're particularly interested in, how these cells in the gut lining control the sensing of nutrients, and then how that sensing is communicated to the brain. And it looks as though this endocannabinoid system is at the seat of this nutrient sensing and then neurotransmission up to the brain. And the endocannabinoid system plays a very important role in these processes.
So when you say nutrient sensing, for example, is it particular kinds of nutrients, for example, like lipids or fats or any kind?
Yeah, it looks to be a variety of different macronutrients. For example, dietary fats and sugars and carbohydrates too, right? Carbohydrates and sugars. These can stimulate release of molecules such as the incretins. Okay. So a variety of macronutrients can stimulate incretin release. So what are incretins? Incretins are released from the small intestine and communicate with the pancreas and aid it in releasing insulin, right? And as we know, there's all of these drugs out on the market now, such as Ozempic. They mimic these natural incretins, such as GLP-1, GIP, and in fact, cholecystokinin is considered an incretin as well. There's another function, however, of these incretins. And they can activate the brain, either directly or indirectly through the vagus nerve. And what we've shown is that the CB1 receptors are uniquely positioned between the sensing of the nutrients and the release of these incretins. And we could talk about this a little bit later, but get a little more in depth in the complexities of this. But CB1 looks to be at that interface of not only glucose homeostasis, but also possibly food reward and how we sense these high energy nutrients and what makes us attracted to them. And that's another aspect of this endocannabinoid system that I think is very important for everyone to understand. I like to think of this as the thrifty lipids system. What does that mean? There was the thrifty gene hypothesis from years ago, and we it was fun with my postdoctoral mentor years ago, Dr. Daniele Piamelli. We generated this fun review article called The Thrifty Lipids, Endocannabinoids and the Neural Control of Energy Conservation. And what we discussed in that, and it sort of encapsulates how I think about and my laboratory thinks about the endocannabinoid system, is that it's involved in the seeking of high energy foods, the sensing of the high energy foods, the absorption of the high energy foods, and then the storage of the high energy foods. Activating this system, at least acutely, can drive all of these processes. Why would this have been evolutionarily conserved? Well, it's feast or famine. We haven't evolved these systems out of our body, right? It's still, our bodies are set up for feast or famine. It just so happens that it's feast all the time, and we don't run through, in large parts of the world, too many famines. So the system sort of becomes hyperactive over time and drives us to go eat at McDonald's. And it's so easy to access all these calories, where before we'd be running around in the savannah searching out high energy nutrients. tasting them. And then, boy, once we taste them, it actually triggers the endocannabinoid system in the gut. It makes us want to eat more of them. So it's a pro-survival system that in modern times can lead to overeating and obesity and other metabolic disorders such as type 2 diabetes.
So along those lines, a lot of people think of overeating as mainly about willpower. Your work suggests there's a lot more going on with gut-brain signaling.
Yeah.
And that could promote overeating. Tell me more about what your studies show.
Yeah. Yeah. So as we just described there, this sort of pro-survival system can override normal homeostatic feeding, right? We go about our day. We want energy balance. We generally don't overconsume or underconsume until we have access to those high energy nutrients. So what we found is that in the gut lining, the cannabinoid receptors, so these are the locks. Remember, the endocannabinoids are the key that fit these certain locks. These locks are on most of the cells in our body. And the endocannabinoids are produced by other cells or the same cells as well. And these wind up activating cannabinoid CB1 receptors, right? So what we found is that in diet-induced obesity, so we use mouse models, and mouse models are very important to get at the mechanisms, cell-to-cell communication, for example. What are the mechanisms involved in this signaling? We can't do those sorts of experiments in humans, obviously. So these are mechanistic studies where we begin in rodents. So we've been looking in rodents and using a diet-induced obese mouse model where we feed the mice a Western diet, right? So a Western diet is comprised of high fats and high sugars, and that basically just represents the amount of the composition of our diets in the Western societies. Although it's not Western anymore per se, it's throughout the entire world at this point. So there's this drive to consume these sort of high energy nutrients. The animals, mice, when you give them access to the high fat, high sugar diet, mind you, it's matched for the average consumption and average macronutrient content that humans consume, such as 40% of their calories come from fat. So we match that. What we find is the animals love this stuff. They overeat. It's almost like they're binging out on this food, almost like drug taking behavior, in fact. They're binging. They love this stuff. They rapidly overeat. They become obese rapidly. What happens is in the intestinal lining of all places, the endocannabinoid levels spike. They go up really high. And what we found over the years is that this, in turn, is driving this overconsumption of foods. For example, how do we know then that it's cannabinoid receptors in the gut? Most of our studies, for example, are pharmacological, and they're wonderful and very important studies. The issue with administering a pharmacological agent, as we know, is that it's systemic, and it activates cells or inhibits cells if it's an antagonist all throughout the body. Now, while that's a nice first step to identifying how systems work in the body, it doesn't get very selective for certain organs. So we've developed transgenic mice in the laboratory that are in fact missing that lock that endocannabinoids bind, that lock that THC sort of rents for a little while. We can selectively remove those receptors from select organs. Now we can start to look at what does that endocannabinoid system do in one particular organ system. And what we found is surprisingly, when we remove the endocannabinoid system, so just the receptors from the intestinal lining, we give the mice, for example, a whole lot of behaviors are different, but in this context, for this discussion, if we give the mice a preference test and we say, do you rather this high fat Western diet, high sugar Western diet, or just a cruddy old rodent pellet, low fat, no sugar? They love, of course, the high fat, high sugar diet. Normal mice, when you knock out that receptor in the gut lining, however, you give them the preference test, they're indifferent. That's telling you that this system in the gut lining is critical, at least for acute preferences for these high energy diets. Something else that's interesting when you look at these mice, they have a slightly lower body weight gain over time when you give them a Western diet, and they consume roughly the same amount as a lean mouse. So this system is promoting the overconsumption of diets that are high in fat and high in sugar. This is the endogenous system. Well, that's in the gut. So we may be thinking, what about the brain, right? I'm a trained neuroscientist. What the heck do we even care about the gut? Well, there's communication that goes on between the gut and the brain. So our gut is through the autonomic nervous system. And it's really without awareness is help guiding our behavior throughout the day. In fact, telling us when to eat, when to stop eating. And we found that this system in the gut lining is very likely controlling that neurotransmission back and forth from the gut, called the vagus nerve. We found that the cannabinoid system is controlling how the signals get from the gut to the brain. So for example, normally you eat a meal, comes into your intestine, it's sensed, and you release molecules, such as those incretins that we talked about before that can help with glucose homeostasis. But what they also do is tell you to stop eating. So if you're eating there, Igor, you're having your sandwich, and I come and inject you with cholecystokinin, for example, it's released from those cells. You would say, you wouldn't feel sick, but you would say, Nick, you know what? I'm feeling full. That's a satiation peptide. What we found in diet-induced obesity, overactivation of the endocannabinoid system, which we found blocks the ability for the gut to communicate with the brain and tell the brain to stop eating. So we can go in there and tweak this system and improve overeating and perhaps food preferences in rodents, as well as hopefully in the future in humans as well.
So going along the lines of, using your mouse models to knock out or get rid of certain receptors, as an example, in certain organs, are you saying that if we had marijuana users who were knocked out for CB1 in the gut, they wouldn't feel the munchies? Or
well, this is, the billion-dollar question. If we could only recruit folks to let me inject the virus into their intestine. Well, theoretically, the answer would be yes. We still need to test this in humans. So, of course, mice are mice. Mice aren't humans. And as I mentioned before, the rationale for using mice is that we can dive deep into the mechanisms of what's controlling these physiological processes. And what can wind up happening, for example, we try and mimic human conditions as well as possible. So as I mentioned, we give rodents access to this Western style diet. It's high in fats, similar amount average to what humans consume, and high in sucrose. So we try and match that to humans as well as we can, on one hand. On the other hand, our mice are in such a controlled environment, and we do that on purpose. They're almost genetically identical, the mice, how they're bred. They get the exact same amount of food each day, the exact same temperature, humidity, lighting. Well, all humans aren't under those exact conditions. So we have to recognize the limitations to rodent studies and let these inform human studies. There's one other component that I'd like to point out about the validity of certain models for the human condition. So we'll talk about cannabis then and how it can impact these systems. So we use cannabis to impact these systems within the body. But how do we know that the levels are similar to what's found in humans? So we try and match the amount and standardize the amount of THC, for example, that's given. So that primary psychoactive, the intoxicating component of cannabis, we give 5 milligrams per kilogram of THC to a mouse. What's been found is that dose leads to the same amount of THC in blood in humans after smoking roughly one marijuana joint, as administering 5 milligrams THC to animals. So we try and again mimic the conditions the best we can. But of course, we have to acknowledge that mice are mice and are not humans.
Yeah. So 5 milligrams per kilogram for a 70-kilo person would be 350 milligrams of THC. I think people would definitely feel that dose.
Yeah, no, exactly.
They might even be kind of unconscious after it, right?
Well, yep. Yeah.
But I think your point is also we have to take into account that the metabolism of, for example, rodents is very different than that of
it's very
humans.
high. Yep.
And so it is.
Yeah, we're metabolizing them differently, huh? But at the end of the day, it leads to the same amount in blood.
Right, and that's interesting. Right. So, can we talk just a little bit more then about when you introduce cannabis? I mean, you've talked about the endocannabinoid system and how it's part of this whole regulatory framework
Mm-hmm.
governing, food-seeking satiety and so forth. What happens when you introduce artificially then cannabis into the system, which is what happens with people?
Yeah, exactly. Yeah,
How
so
do
for
these signaling pathways change as far as you can understand it?
yeah, right. So we recently put out a rather large study examining this exact phenomenon. So it's known and has been recorded for thousands of years that cannabis can stimulate appetite and increase hunger. This has been documented for 2000 years BCE. And so we term this now the munchies, right? Everyone that's ever consumed cannabis understands what the munchies are. It's making you probably eat more than you normally would during that moment when you're intoxicated.
Particularly some of those foods you mentioned.
Uh-huh.
You didn't mention Doritos specifically.
Yeah. Well, I can promise you it probably would work on Doritos. Yes, exactly. So it kind of drives you to overconsume these foods called the munchies. There's something that I like to call, however, the cannabis paradox. And this is really surprising. So chronic cannabis users weigh significantly less than non-users, and in fact have a reduced probability of developing type 2 diabetes. Seems paradoxical. If you're overeating and munching out on lots of high energy foods, storing more of this energy, you'd be gaining weight, increasing the probability of glucose homeostasis, dysregulation of glucose homeostasis. So this was the exact problem that we wanted to attack in one of our latest studies in the Journal of Physiology that was led by graduate students in my laboratory. So what we did is we asked the question to these mice, the best way we can ask a question. How does cannabis impact body weight, glucose homeostasis, as well as expression and function of a variety of signaling mechanisms in the body, including how the adipose fat tissue, in fact, talk to the islets in the pancreas. And so we designed these experiments to, again, try and mimic the human condition as best as possible. So we had several groups of mice. One of the groups was a control group where it was just fed a lean diet. Okay. They maintain normal blood glucose homeostasis. If you give a glucose tolerance test, they have a nice little spike in blood glucose and then it comes back down. The body's absorbing it. If you give mice the Western diet, okay, they have impaired glucose clearance. There's much higher levels of glucose. They're hyperglycemic, hyperinsulinemic. This model represents really pretty well the human condition of type 2 diabetes, right? So we have these groups now, and we administer different compounds to them. Okay, so remember, the obese mice are hyperglycemic, have impaired glucose clearance. So we wanted to ask, how does cannabis impact these systems and their body weight? So what we did is we had side-by-side groups. We used that 5 milligram per kilogram of delta-9 THC, again, which leads to very similar levels in humans after consuming one cannabis cigarette. The problem with delta-9 THC is there are very few people just consuming delta-9 THC out there. They consume a whole cannabis product, right? Which has a whole host of phytocannabinoids, hundreds of them. So we wanted to match that condition. So what we did is we got cannabis again from the NIDA drug supply program, and we did our own extraction, a cold ethanol extraction to get all the oils out, all the cannabinoids out of the plant. We wanted to match THC levels. So one obese group gets just THC, and the other group gets THC, but all the other chemicals at the exact same amount. So we run it through our mass spec. We use UPLC mass spec in the laboratory to identify these chemicals and very accurately measure the amounts of the chemicals in the plant. So we wanted to match pure THC, what's its effect, and then all cannabis extract. I can give you a quick anecdote how this wasn't very straightforward. We extracted all the chemicals out of the plant, and I run it through my mass spec, right? My crew runs it through. And we look, and there's barely any delta-9 THC in this cannabis. So I'm starting to get a little worried. I say, wait, is something wrong with this cannabis? This is some bum cannabis from the government, right? It has no THC in it, right? So then we started thinking, well, cannabis is combusted. right? So it's heated by combustion or heated, put into oils and made into different foods. That process of heating converts an inactive form of delta-9 THC, which is in the plant. So mind you, if you took a bud off of a plant and ate it, it's not going to really do anything. There's not active delta-9 THC in it. You have to heat it. So we optimize the temperatures to do this full decarboxylation to basically convert the inactive acid form of delta-9 THC into active. And I'm proud to say we were 99.99% conversion of acid version over to pure delta-9 THC. So we had some technical challenges. Once we got this all sorted out, we now administer these compounds to the obese mice. So those that got delta-9 THC or those that got delta-9 THC, the same amount, but all the other chemicals lost body weight. Those obese mice became lean, almost down to the level of the lean mouse body weight. So we said, wow, this is cool. Okay. Same exact amount of body weight. So now let's look at a whole variety of other parameters. For example, glucose homeostasis. So we did glucose tolerance tests, insulin tolerance tests. And these enable you to be able to look into the body and say, how well is the body processing glucose? How well is that system working? How bad is the insulin resistance, or how petered out are the beta cells and able to secrete out insulin? So we run these tests and we find, curiously, despite that body loss, the mice that were given just purely delta-9 THC, they were still diabetic. There was no change in glucose clearance. However, the mice that received the whole extract with THC and all the other chemicals completely reversed that diabetic phenotype. And the mice responded like lean mice at that point. So we began to look into what mechanisms may be involved in these processes.
So are you saying in effect that it is some of these other cannabinoids, there are other chemicals there called terpenes, of course, that constitute what some people have called this entourage effect? Is it those things somehow in combination with THC that are doing the trick, or what?
Yeah, excellent question. The $1 billion question again here, right? So we're not sure yet, but we're actively and systematically going through different subcomponents of cannabis, whether alone or in combination with delta-9 THC that may be leading to that pro-metabolic effect and the reversal of type 2 diabetes. So this is an area that we really want to pay attention to in the future. We're trying to as well gain extra funding to really go through each of the different chemicals. But what we can say is that THC alone will lower body weight, but you're still, at least in the mouse model, will be diabetic. There's some other component, either alone or in combination with THC, that may improve glucose homeostasis. And like I said, we're going to be systematically going through those chemicals. So stay tuned.
Good. Well, actually, that leads us naturally to my final question, which is looking ahead, what would an ideal therapy look like based on your research? Would it be targeting reducing appetite? Would it modulate satiety, improve metabolic health, all of the above?
Yeah. Well, all of the above. As we know, rimonabant was on the market, in fact, in Europe as a Complia. So rimonabant, as we know, is this CB1 selective antagonist with inverse agonist properties. And it was developed by Sanofi Aventis and worked wonders, reduced waistline, improved metabolic function. However, when you started to pour through the data, there was increased psychiatric side effects from this drug. In hindsight, to no real surprise, because you're taking a drug that's systemic. We know the systems in the brain in areas that control mood, emotion, anxiety, and stress. So that drug was crossing into the brain and disrupting neural activity, which can lead to these psychiatric side effects. So what I think is very exciting about our work is that we've targeted an area in the periphery, and in particular, the gut lining, that can be potentially therapeutically manipulated for therapeutic gain in humans to improve metabolic syndrome. Now, there are several molecules already going through phase 1B, phase 2 clinical trials. A couple of them are peripherally restricted CB1 receptor antagonists, and for the treatment of metabolism. So we'll see how well they do, and if they can go through all of the trials. There's one.
Just if I could clarify, when you say peripherally restricted, you mean they don't get into the brain, right?
Yeah, they're tweaked. You can almost think of it as rimonabant, and it's tweaked just a bit so it doesn't cross into the brain very readily. There's an issue, though.
Something that is affecting the rest of the body, but not
Exactly.
the brain.
So it's a way to be able to exclude, presumably, a lot of the psychiatric side effects by just manipulating the system in the periphery. That's enough to have metabolic gain without those side effects. And so we call these peripherally restricted CB1 receptor antagonists. And that, again, they're largely restricted to the periphery. But again, they're bathing the whole body. So from a scientist perspective and mechanistic perspective, it's still bathing the body. So we like to look at knockouts to be able to see exactly what the system's doing there. But what we believe is we can remotely control the brain from the gut. And these potentially new molecules could have therapeutic benefit without those bad psychiatric side effects. One thing that happens in humans that's known in diet-induced obesity, you can get a slight disruption of the blood-brain barrier. So you still need to be a little bit careful even with peripheralized CB1 receptors in humans, because they may be able to access a bit, but access the brain because of this compromised blood-brain barrier. Future therapeutics, and which we're working on, would be to target specifically in the gut and not have them even go systemically within the periphery. Well, A, they're not going to go systemic, but B, not even in bathing all the organs in the periphery, just in the intestinal lining. I think this would be an area to look at for the future, as well as all these minor components that may be involved in this either in combination or alone with delta-9 THC that can reverse this diabetic phenotype. So it's a really exciting time for cannabinoid research right now, and it's a fun area to be in, and I feel just very grateful to have been in the past 26 years now working on this system. It's been fun.
Yeah, it's been quite a journey from the early days, 3 or 4 decades ago, when Raphael Mechoulam was first looking at, first of all, what is the actual active ingredient, and then the discovery of these receptors and the internal signaling systems. We've come a long way to seeing that these are important additional ways that control not just our behavior, but also our physiology. At a very basic level. Well, we're coming to the end of our time. Is there anything else you wanted to say by way of wrap up?
Yeah, we need to support research. Please, everyone, contact your senators. Everyone, we need to support research because this is where cures come from. So that would be my big takeaway. Just please, let's support research because this informs medicine.
Well, that's an important lobbying statement. Good. All right. Well, Nick, thank you so much for taking the time and speaking with me today. You've really opened up this whole area of gut feelings, right?
Yep. My gut made me do it.
That they're really there, and there's a whole mechanism for that.
Yep. Yeah, it's my pleasure. This was a lot of fun, Dr. Grant. Thank you so much.
If you like this podcast, please subscribe so you don't miss future episodes. For more information on the Center for Medicinal Cannabis Research at UC San Diego, please find us on social media, including Twitter/X, Instagram, LinkedIn, and other social media platforms, or visit our website at cmcr.ucsd.edu. Thanks for listening.